Yeah, welcome back, students. So, in this study, in this video, I'll be discussing about mutations studies in Drosophila or mutations in case of Drosophila. So, first thing is we have to know why Drosophila. So, as we all know very clearly, Drosophila is the important genetic model, or you can say model system for genetic studies. That's the one main reason. Second is it is called as the central of genetics, as we have large number of mutants that can be studied, and all the mutants are simulated with respect to human nature. Okay, either physically or with respect to specifically with respect to the physiological aspects are simulated with respect to human beings, and they are studied. Hence, Drosophila serves as a model organism, and hence it is referred as the central law of genetics, as we all know about it well. Okay, so there are immense and very different types of mutants which provide us with respect to different types of inheritance pattern, Mendelian inheritance pattern, Morgan's inheritance patterns. Okay, so and linkage mapping. So there are n number of applications which can be utilized by knowing very well about Drosophila. Okay, so before going to Drosophila, we have to know what is a mutant, what is a mutant phenotype. So that is the main aspect of Drosophila. Even in your people's practicals, we have about this. So each and every observable character is called as the phenotype, and each phenotype is controlled by a gene, which is further expressed in the form of a function. Okay, and that can be seen either in the form of a protein or in the form of a character or behavior character or anything like that. Okay, so always these genes are under the control of expression and penetrance, and results in the way in which they penetrate to express themselves, forming different levels of phenotype. So there is no one type of phenotype. It's a gradual expression based on the allelic expression. We consider the mutant phenotype, and that is in turn connected to the mutation which is holding it. So hence a mutation means it's a change, as we know very clearly, which undergoes, and these different types of mutations have different effects of phenotypes, and they have different mutation rates. Okay, in any species, including humans. Sometimes they may be carrier, sometimes they may be expressed. Okay, so we have many type of characters which are directly observed, which may not be observed. Okay, and they are connected to the other organism in one or the other way. So what we do, we simulate those type of mutations in case of other organisms and see how exactly they behave there, and we try to simulate that to the human beings. Okay, so that's why we use this one organism, that is the Drosophila, for the genetic studies relating to human beings. So what is so special about Drosophila? Drosophila is a genetic system, as we know very clearly, which was used by Morgan. So we have a number of mutants. About 4,000 genes have been observed with respect to various alternative types by Lindsley and Zim in our lab in your college. We have this book, Lindsley and Zim, Drosophila monogaster by Lindsley and Zim. Anybody who is interested can go and have a note of it. A very important book. Okay, so from that book we will get all the information necessary with respect to Drosophila's genetics, development, behavior, cell biology, all the things we can learn. And today Drosophila has become a very important aspect because of all this reason. Okay, so we can engineer it, and we can get desired combination easily by genetic recombinations. So, and we can map and test them and find and relate hypothesis, develop hypothesis and find the results. So as for research purpose and all, we use it. Okay, so I'll describe here the different types of mutants in case of Drosophila. So when we hear the name Drosophila, we get variants. Okay, so one of the most important variant we try to understand in case of Drosophila is the I mutations. So even in case of human beings, when we take the I eye color, there is range of eye colors we can see. Sometimes that may be correlated with respect to the skin color, but most of the time it is the retinal, or you can say the pigmentation which is there. So here you can see very clearly with respect to Drosophila, uh, white eye color. Okay, so this is white, very clear white we can see, and there is lightly white color. So there is a pattern which is getting degraded, the color. So it is multigenic in nature. Okay, as the number of dominant allele gets increased, the color becomes more and more dominant from white to the sepia. Okay, so there are 15 different colors we can see here. There are about 8 different colors which are shown here. So this is also an example which we study with respect to multigenic, but it is also said that it is single genic, but single genic character, but it is related to one expressive pattern. Okay, because as the number of dominant allele increases, the color domination also increases in case of Drosophila eye pigments. So if you see, these two can be easily identified. One is white eye color and the other one is the sepia eye. Okay, in between that there will be a normal red eye colors, but here you can see cinnabar, vermilion. Okay, so when we see Drosophila exactly, we will come to know very clearly, but it's a photo, it's not so clear with respect to rest of the colors. Next, the same way, one more important Drosophila mutations which we study is with respect to the wings of Drosophila. Okay, so in your laboratory, of course, we had to show in your practicals, but this time it is not possible. So we will see here that there are different types of Drosophila mutants, which as well as orientation on the body, you can see the different types of mutations with respect to the wing types. Okay, so extremely reduced, small, cut wings, okay, reduced wings, wingless. So like that, different types of mutation, Drosophila wing mutants are also available for genetic studies. Next most important character is the body color. So you can see here it is the yellow body, it is the sepia body, okay, so means the black body, and the other one is the shining black. You can see like the normal house flies. Here there is a type of fly which is observed here, but it is not the house fly, it's a shining body. And the same way we have white bodied also. White eye is there, and it is white eye in the same way. Sometimes the paleness increase is observed in the body also, but most importantly we go with yellow body and sepia body and the black body. Okay, ebony body can be observed. So these are the type of mutants which we have in our labs, so which can be used for crossing and finding the results. Then comes the multiple mutations. As I said, sometimes that will be having normal winkle. No, there will be having yellow body with vestigial wings. The same way, sepia body with vestigial wings, or wingless with white eye. So like that, there are multiple mutants, or you can say mutations present in the same Drosophila. So these are called as multiple mutants. Okay. So the question probably may be asked is in practicals. You have to be very thorough with this, because either in Viva you may be expecting this question, or we have to study and write with respect to the spotters. So the question probably is the write a note on mutations in case of Drosophila. So here, to answer this question, what we have to do, we have to introduce why Drosophila is referred as central of genetics, its significance with respect to human mutations, types of mutants available for genetic studies. So if we are able to congregate all these ideas, means we will be getting good marks. Okay, so remember, if the question is asked in the examination. Thank you all.